Alright, so continuing on with our photosynthesis unit, we're going to delve into the light-dependent reactions in this screencast. We're going to look at the reactants, we're going to look at the products, the implications of both, and how these are so incredibly important for life on Earth and for our continued existence. So let's get right into it. We'll look at the process. First, we need to talk about where it takes place. We need to talk about within the chloroplast itself, and here's a graphic representation of a chloroplast. You have these stacks of thylakoids, of course. One stack is a granum, so here's our stack of thylakoids. The mechanisms for the light reactions are embedded within this thylakoid membrane. The photosystems we're going to be talking about are embedded in this membrane. It's a phospholipid bilayer, just like all the membranes we've already talked about. And it has pigment molecules in the photosystems of this process, and they are the molecules that are going to trap the light energy and allow plants to turn it into the energy in the bonds of carbohydrates. So just keep in mind, this is where we are. Okay, we're in the thylakoid membranes. The next process, the light-independent reactions, are going to be taking place out here in the stroma surrounding the thylakoids. But for now, we're gonna stay in here. The big picture, like we said, here's our thylakoid membranes. Here's our light-dependent reactions. What do the light-dependent reactions need in order to occur? Well, they need water. As you can see, the graphic is showing you an input of water here. It's also showing you, and this makes sense, this is something you surely already knew, that they need light. That's why they're called the light-dependent reactions. What do they release? What are their products? Well, input of water and light. Output, we have a very useful waste product here in a molecule of oxygen. We have adenosine triphosphate, the energy currency of not only plants but our body as well, and a coenzyme, NADPH. NADPH stands for nicotinamide adenine dinucleotide phosphate. Not going to make you memorize that, okay? We can know this as NADPH, a coenzyme. Where do these two very important molecules go? Well, they take their energy to the Calvin cycle, which is the light-independent reactions, and that'll be the subject of the next screencast. So the goal here, we want to see how water and light are utilized to make oxygen, ATP, and NADPH. You're going to see that oxygen is a waste product of plants. They don't use it. It's kind of inconsequential for them, but obviously very, very important for us. So ATP, NADPH, and oxygen, how are those things produced? Well, a lot of it, well, most of it involves redox reactions. All right, redox reactions involve the losing and gaining of electrons. You can remember it this way, okay? Oxidation is a loss, oil rig. Oil oxidation is a loss of electrons. Reduction is a gain of electrons, oil rig. Remember it that way. And chlorophyll is our main pigment. We talked about other accessory pigments, but chlorophyll A is our main pigment. There's chlorophyll B. We talked about carotenoids and anthocyanins and xanthophylls, etc. But the initial energy input is from the sun, and it's going to be taken in by an antenna complex, which is a whole bunch of pigment molecules in the thylakoid membrane, and that energy from this photon of light is going to be funneled to a reaction center. And in that reaction center are chlorophyll A molecules. And those chlorophyll A molecules get excited by that photon of light. And that excitation causes them to lose an electron. Electrons get bumped from low energy states to an excited state. Excuse me. And when they're at this excited state, energy is released. All right. If there's nothing up here to gain this electron, chlorophyll will actually fluoresce. It'll actually glow. Okay, so you can see that. But in a functioning plant, there's going to be a primary electron acceptor here to take this electron and use its energy. So redox reactions are key in this process. We're going to be losing electrons from chlorophyll, Down here at the ground state, they're excited, and their energy is passed on. So let's look at it. Okay, this is our thylakoid membrane. And as I said, it's a phospholipid bilayer. The inside is the lumen, or the thylakoid space. The outside is the stroma. We're talking about non-cyclic electron flow, which means obviously that it's not a cycle. It's moving in one direction. And also, there's a process that's very important built in here called chemiosmosis, which is how ATP is produced. So this is kind of for perspective. We're going to be bouncing back and forth between this slide and the next one, just so you can understand these processes. So here we are. I like this graphic a lot. I think it shows pretty clearly how the flow of electrons occurs between the two photosystems. We talked about there being two photosystems, photosystem 1 and photosystem 2. The reason they're called that, well, first of all, photosystem 1 was discovered first, so they called it photosystem 1, obviously before they knew there was a photosystem 2. But also, photosystem 1 occurred first in plants evolutionarily. So a couple reasons for that. Here we have photosystem 1 on the right. You can see it's called P700, and photosystem 2 is P680. That refers to the wavelength of light that it best absorbs. Okay, we have photosystem 2. These little green particles, these are pigment molecules that are going to be absorbing the light molecules. So a lot of times when you hear this being explained, you'll hear it taught as photosystem 2 first, and then photosystem 1. It really doesn't matter which order you do, but I'm going to start with photosystem 1 since it was first evolutionarily, and maybe you'll see why. We start with this antenna complex, and this is a complex of molecules, of pigment molecules that absorb light. And here comes our photon of light. This energy gets funneled to our reaction center, 2 molecules of chlorophyll A. We talked about the excitation of those electrons. Okay, so here goes that electron being bumped up to a higher energy state. If this primary electron acceptor wasn't here, we talked about how that would fluoresce, it would glow, but it is there. So this primary electron acceptor grabs that electron and starts kind of a chain reaction. Okay, it starts to pass that electron down a series of steps, and as the electron is passed, energy is lost, but that energy can still be utilized. So in photosystem 1, the primary electron acceptor is going to pass this electron to ferredoxin, which is another protein complex that's embedded right here in the phospholipid bilayer. From ferredoxin, those electrons are passed to an enzyme called NADP reductase. They're actually passed to NADP, and that process is sped up by an enzyme called NADP reductase. This is super important. We were talking about, well, how is ATP produced? How is NADPH produced? This is how NADPH is produced. Ferredoxin passes those electrons. It reduces NADP+, a coenzyme. It's kind of a distant relative of B vitamins. Okay, passes that electron to NADP to make NADPH plus H. Actually, it's 2 electrons. But that process is sped up by the enzyme NADP reductase. So now we have NADPH. Let's go back. This reaction center in photosystem 1 lost its electron. It's now electron hungry. Okay, it's absent this electron. If it was a battery, it's dead. So we need to recharge it. We need to somehow replace this electron. How are we going to replace it? We're going to use photosystem 2. We're going to use photosystem 2 to replace the electrons lost from photosystem 1. So same idea. If we move over here to photosystem 2, light photon comes in, trapped by molecules of pigment. The energy is funneled to the reaction center. That electron's excited. It's bumped up to the primary electron acceptor. Once again, here we are. We're built into this phospholipid bilayer. The primary electron acceptor passes that electron, Here we pass it to ferredoxin. In photosystem 2, it's being passed to a protein complex called plastoquinone. All right? Plastoquinone then passes it to a cytochrome B6F complex, a cytochrome complex. Remember, as electrons are being passed, it's going down a gradient. Energy is being lost, but it is utilized. We'll talk about that. Plastoquinone to cytochrome, cytochrome to another protein called plastocyanin, and finally plastocyanin passed that electron right back here to replace the electron that was lost, and now photosystem 1 is recharged. So photosystem 1's happy, it's been recharged. Wait a minute, what about photosystem 2? This electron was knocked out. How does it get replaced? How do we recharge this battery? Well, water is located in this thylakoid space. Water is in the lumen. Okay? This reaction center is so electron hungry that it splits water and it robs those electrons from the water molecule, a process called photolysis, or the breaking with light. So water's broken, it replaces the electrons. What's left over? Well, for one water being broken, there's one oxygen. When you break two water molecules, we have two oxygen, which is O2, which is atmospheric oxygen, which is released into the atmosphere. Okay, so there we are. Now we've seen how NADPH is produced, all right, by NADP reductase and ferredoxin. We just saw how oxygen was created. How about ATP? How about ATP? Well, ATP is produced through a process called chemiosmosis. So we just saw that water was split. We saw that the splitting of water creates oxygen. But what else is left when water is split? Well, we have hydrogen ions, right? Because water is H2O. So we have these hydrogen ions, and they're going to start building up in this thylakoid space. They're going to start building up in the lumen. It creates a kind of an electron gradient of a high concentration in here and a low concentration of hydrogen ions out in the stroma, as you can see right here. This concentration gradient is compounded by the fact that when electrons are passed from the primary electron acceptor of photosystem 2 to plastoquinone to the cytochrome complex, remember, right here, okay, it's just a different drawing. I'm showing you two different perspectives here. When they're being passed down this chain, cytochrome complex starts to pump hydrogens into this thylakoid space. There they are, being pumped in. So not only do we have a buildup of hydrogens from the splitting of water, we have a buildup of hydrogens from the pumping inward of this cytochrome complex. So we have a high concentration, low concentration. What do they want to do? They want to diffuse, obviously. They want to move to a lower concentration. Well, there's only one way they can do this, and it's through this enzyme, ATP synthase. And here we go, chemiosmosis. Okay, this is chemiosmosis. Chemiosmosis is the phosphorylation, or the addition of a phosphate, to ADP using a hydrogen gradient. The phosphorylation of ADP using a hydrogen gradient. Well, the hydrogens want to leave, so they leave through this ATP synthase. And the ATP synthase, as hydrogens leave, it cranks, it turns, and it adds a phosphate to ADP, which is adenosine diphosphate, to make ATP, adenosine triphosphate. All right, so there's our chemiosmosis, our photophosphorylation, or the phosphorylation of ADP using ultimately the energy from light, photosystem 1 and photosystem 2. Now we have ATP utilizing that gradient. Just review. Okay, we showed how oxygen was produced as a waste product from water splitting to replace electrons in photosystem 2. We showed how NADPH is created when it's reduced, or NADP plus is reduced by ferredoxin. And we showed how ATP is produced in chemiosmosis through the buildup of hydrogens in the lumen. Why is it important that we make these? These two specifically will run the Calvin cycle. Okay? Let's look at the Calvin cycle briefly. Don't freak out here. Okay, we're going to do another screencast on this. But look, here's the Calvin cycle. Ultimately, we want to make a sugar. How do we do it? Well, we need ATP here. We need ATP here. We need NADPH in this process. So we have ATP being used in two different instances, NADPH used in one. That means that this process, as it's spinning, is going to run out of ATP faster than it's going to run out of NADPH. So at certain times, this plant is going to switch from being in non-cyclic electron flow to cyclic electron flow. Cyclic electron flow only makes ATP. So when it's starting to run low on ATP, there's going to be a switch that's flipped, and it's going to move into cyclic electron flow, where the ferredoxin, instead of passing its electrons to NADP reductase to make to NADP plus via NADP reductase, the ferredoxin cycles them back to the cytochrome complex to pump hydrogens in to fuel chemiosmosis to make ATP. Notice, only photosystem 1 is being utilized in cyclic electron flow. Photosystem 2 isn't being used. Only ATP is produced because the ferredoxin pumps back to cytochrome, okay, brings the energy here for the chemiosmotic processes, instead of NADP plus. All right? So those are the light reactions as a whole. Hopefully that makes sense. Hopefully it'll make more sense once we get through the Calvin cycle and we can really see the big picture.